Welcome to Pharmaceutical Calculations, a Conceptual Approach, Lesson 7, Concentration Units in Pharmaceutical Sciences. Our today's objectives is to discuss the difference between density and concentration. As a reminder, density and specific gravity were extensively discussed in Lesson 6. Then define solute, solvent, and solution, and finally go through some of the concentration units commonly used in pharmaceutical sciences. Now, we're all familiar with the equation expressing the density of a material. And as you can see here, equation 4.1 looks identical to the formula for the density. Concentration is also written as mass over unit volume. However, these two properties are very different. The main difference is that between the two is that density refers to the mass of a pure substance present in a unit volume of that same pure substance. For example, the grams of water present in 1 ml of pure water. Whereas concentration is defined as the amount of substance present in solution or solvent. Solvent is a different substance than the substance dissolved in a solvent. And of course, solution is the mixture between the solute or the substance dissolved in a solvent and the solvent. So this is the main difference between the two. Now, there are a couple of other more subtle differences. For example, concentration is the amount of substance present or dissolved in a solvent or a solution, and this amount could be written as mass of volume. Whereas density is always the mass of the substance over the volume of the unit substance. We do have concentration units in pharmaceutics that express the volume of a substance dissolved in a volume of a solvent, for example. One more difference is related to the dependency of density on the temperature, especially if the temperature range you are working in is very close to the phase transition of the material. We are all familiar with the three states of matter: solid, liquid, and gas. The density of liquid water is 1 gram per ml. The density of ice water is 0.92 grams per ml. So ice water is lighter than liquid water. And water is one of the few molecules that the density of its solid state is less than that of its liquid state. And it is actually due to that property that aquatic life can be preserved during the cold days of the winter. And I'm referring to the lakes here, where ice flows on the surface, leaving liquid water beneath and the aquatic life in it. Okay, so these are the main three differences. Now, concentration is not always dependent on the temperature because in some occasions the concentration unit is mass of a substance over the mass of the solvent or the solution. We're going to see some of these examples in a minute. Now, let's move to the definition of solute, solvent, and solution. Solute is the substance that is dissolved in a solvent to form a solution. It could be a solid, liquid, and very rarely a gas, and I'm referring to anesthetic gases in pharmaceutical sciences over here. Solvent is the substance that dissolves the solute and is usually present in the greatest amount. It could be solid, liquid, or semi-solid. We are more. All three exist actually in pharmaceutics or in pharmaceutical sciences, although we are more familiar with liquid solvents. We're going to see some of the differences soon. And I guess here we're going to see the differences. So solution is a homogeneous mixture consisting of the solute and the solvent. What we need to understand here is that solute dissolves in solvents because of chemical interaction. Now, when the solvent is solid or semi-solid, it is expressed in units of mass. When it's a liquid, it's expressed in units of volume. Now, some of the examples of solid dosage forms and semi-solid dosage forms. So solid dosage forms are powders, capsules, and tablets. semi-solid dosage forms, creams, ointments, and gels. And I've written here that the solutes are uniformly or homogeneously dispersed in these media, solid media or semi-solid media, right? And there is a difference between dispersion and dissolution. A solvent dissolves the solute by chemical interaction. On the other hand, a solute is dispersed in these media without any interaction. For example, Vaseline, when you make a cream or an ointment, Vaseline provides for the dispersion of the solutes, but it doesn't really chemically interact with them. Now, if the mixture is heterogeneous, there is no concept of concentration because the solute is not uniformly dispersed throughout the mixture. Let us now take a look of some of the common concentration units in pharmaceutical sciences. First one, mass per unit volume. We don't need to say anything here. Common units, grams per ml, grams per dl, and so on. Percent by weight, written as percent W over W, it's quite common in pharmaceutical sciences and is quite commonly asked in exam questions. So pay attention to this. Essentially, it's the grams of solute in 100 grams of mixture or solution. Two examples we have here is betamethasone ointment, 10%. Some people, because ointments is semi-solid, it's implied that is weight per weight, and they don't write weight per weight. I would encourage you to write it anyway. So this means 10 grams of betamethasone over 100 grams of mixture, betamethasone included. Now this unit, percent weight per weight, is also used in chemistry, especially to express the concentration of strong acids and strong bases. And here I cited an example, HCl 37% weight per weight. It means 37 grams of hydrochloric acid dissolved in 100 grams of solution. So now here, although the HCl 37% is a liquid, for whatever reason is expressed in units weight per weight. When you use it, because it is a liquid, you need to transfer a certain volume. In order to calculate how much volume you need to transfer, you're going to have to convert these concentrations in weight per volume. And in order to do that, you need to know the density or the specific gravity of the hydrochloric acid 37%. Now, when do they actually use this weight per weight for acids and bases? Usually there is no rule, but we see it when the concentrations of acid and bases are higher than 10 molar. However, there is no rule. Just read the label, and if it is percent weight per weight, use the density and specific gravity to convert it as weight per volume. We're going to see how to do that anyway. But this conversion from weight to weight per volume is very common in exam questions, even license, pharmacy licensure examinations. So you have to be proficient in these conversions. Percent weight per volume, this is what I was referring to, is grams of solute in 100 milliliters of solution. Percent by volume, this is the volume of solute, so mLs, milliliters of solute dissolved in 100 milliliters of solution. For example, alcohol USP is a 95% volume per volume, and this is an information that you need to memorize. Alcohol USP is not 100% pure alcohol, it's a 95% pure alcohol. So 5% of it is water. Molarity now. This is a very common unit used in every science, not just pharmaceutical sciences. It expresses the moles of solute per 1 liter of solution. As you can see here, sodium chloride 0.15 molar. This is the isotonic solution. We're going to study isotonic solutions later. Is equal to 0.15 mole of sodium chloride over 1 liter of solution. Notice that when I use units in a formula, when I use moles in a formula, I write it as mol here. Whereas here, when I use it in a sentence, I write it as moles. Of course, these are the official rules. And no student will be penalized because of that. But my suggestion is that write everything according to the rules, because they are there to help us communicate. If you skip the rules and use your own rules in writing scientific texts, at the end we may be unable to communicate. Molality. This is a very different unit, so that's lower letters M. It expresses the moles of solute in a kg of solvent, and because both of them are units of masses, this unit is not sensitive to temperature. And this is not about the chicken and the egg. So density is expressed in mass per volume. So density is sensitive to temperature, but mass is not sensitive to temperature. Mass is not even sensitive to gravity, right? The weight is. So this is the advantage of using these units. It's not as commonly used as molarity, but you may see it from time to time here and there. So you need to know about it, especially in pharmaceutics, in physical pharmacy. When we study colligative properties of solutions, quite commonly osmotic pressure is expressed in molar instead of molar units. When, of course, you're dealing with dilute solutions, both of them approach the same number. Normality. So this unit is still in use still in use in pharmaceutical sciences and clinical sciences. You have to be very careful with it. You need to know what it is, because you use it to calculate equivalence of drugs. So this unit denotes the equivalence of solute per liter of solution. The unit equivalence, this is the unit for the equivalent weight, EW. And it is important to understand that normality is always defined in terms of some chemical reaction. So unless you know the reaction, you won't be able to write down the normality of that solution. For acid-base reactions, the equivalent or the equivalent weight or gram equivalent weight is defined as the weight in grams of an acid or a base required finishing or accepting exactly 1 mole of protons, respectively. So here I have an example. Sodium chloride 150 milli normal is 150 milliequivalents of sodium chloride over 1 liter of solution. Okay, so this is very useful here. Of course, this is a salt that dissociates, let's say 100% in solution. This concentration is high, but we assume that this is 100%. That's why whatever you have there, it all gets hydrolyzed or dissolves or dissociates in solution. Okay. Now, the equivalent weight of a substance, this is equally important, please pay attention, is called the mass of one equivalent, just as the molecular weight of a substance is the mass of 1 mole. So this is the definition of equivalent weight, gram per equivalent. So the relationship between the equivalent weight and the molecular weight is expressed with equation 4.2. Equivalent weight is equal to molecular weight over N, and N here is basically the equivalence per mole. Normality, so if you factor in equation 2 the volume, the unit volume, then you will derive equation 4.3, which is normality equals to N times M. And again, N is the equivalence per mole. It is defined as the number of moles of proton furnished by 1 mole of acid or accepted by 1 mole of a base participating in the reaction. So you, again, I emphasize, you need to know the reaction. Most of us are familiar with this. We are not familiar with equation 4.2, but you better get familiar now, because you're going to use it extensively to calculate doses of electrolytes and other drugs, ionic drugs. Okay. Ratio strength, another rare unit which is used quite commonly in pharmaceutical sciences, and especially compounding pharmacy. So this is a unit that expresses one part of the solute per part of the solution. So I emphasize that because the solute or the numerator has to be equal to 1. If it's a solid, it is expressed in grams. If it's a liquid, it's expressed in milliliters. So solids and semi-solids are expressed in grams. If they're liquids, are expressed in milliliters. Now, it's not all the books that follow this rule. Please read carefully and make sure that the units are written next to the ratio strength. Let's take a look of hydrocortisone cream 1/100. So this is expressed as 1 gram of hydrocortisone per 100 grams of cream, hydrocortisone included. That's the cream ointment, right? Aspirin solution 0.1/100, that's not ratio strength officially, right? You need to convert it to 1 gram of aspirin per 100 ml of solution. So the ratio strength in this case is 1/1000 grams per ml. Remember, the numerator, which is a part of the solute, has to be equal to unity. Okay, very important. So the numerator has to be equal to unity. Parts per million, another unit that is commonly used in toxicology and of course environmental pollution. So this unit, parts per million by mass, is basically it expresses the parts of solute in a million parts of solution. If we are dealing with solids, they are expressed in grams. Parts of liquids are expressed in milliliters. And again, this is the rule that is followed by most of the books, but not everybody is following this rule. Just make sure that you read the units properly. For example, 3 ppm of mercury, what does it mean? It means 3 parts of mercury over 1 million parts of water. That's equivalent to 3 grams of mercury over 1 million grams of water. And because the density is 1, it's 3 grams of mercury over 1 million mLs of water. And because the composition, the amount of mercury is so little, we consider solution and water to be the same density. So basically 3 ppm mercury is 3 grams of mercury in 1 million ml of solution. Parts per billion. So the only difference here is the parts of this solute in a billion parts of solution. So 3 ppb of lead is 3 parts of lead over a billion parts of soil, or 3 grams of lead over a billion grams of soil. And since soil is a mixture, right, it's not a pure substance like water, this is really a physical mixture or a homogeneous mixture here. So here goes the ppb. Now, mole fraction. This is a common unit for chemistry. It's not used so much from pharmaceutical sciences. However, when you study physical pharmacy, especially colligative properties again, you may use that one, Raoult's law, Dalton's law, it's always there. So the unit, this unit denotes moles of solute per mole of all components that constitute the mixture, as equation 4.4 is right in there. So this is the mole of solutes over the moles of all the components composing the mixture, including the solute. Now, the summation of all the mole fractions is always equal to 1. That's always true. Lastly, the activity unit. So this unit is mostly used for proteins, enzymes, but also for other smaller synthetic or natural molecules. So synthetic or natural molecules that are antibiotics or anti-cancer agents, they are also expressed quite often with activity units. Now, the actual definition of the activity unit is dependent on the physiological function of the molecule. For example, if this is an enzyme, the catalytic activity of an enzyme is defined as the number of substrate molecules transformed or converted per unit time by a single enzyme molecule. So that's the definition of the enzymatic activity. Now, if this unit activity involves an antibiotic, so basically the bactericidal activity, it is defined as the amount of bacitracin, for example, needed to kill a certain population number of a particular bacterial strain per molecule again. So the activity depends on the physiological function. Now we have to be careful with the unit of activity as it provides information additional to purity. So for instance, it may happen frequently actually that you would buy the same enzyme from two different sources, two different suppliers, that the specification written describes an identical purity of that molecule, but they do have very different activities. Why is that? Well, there could be two reasons. The first one is that the enzyme from these two sources, or the molecule from these two sources, may be of identical purity, but the conditions under which it was purified and the composition of solutions used to reconstitute them may be affecting their active conformation during the renaturation process. Because remember, you purify these enzymes or these biological molecules and you have them in dry form, and you reconstitute them just before you use them in the clinic. So that reconstitution, composition of the reconstitution solution may be affecting the active conformation of the molecule. Another reason for a different activity, although the purity is written as being identical, is the conditions at which the drug activity is evaluated by the two manufacturers. It may be different. Okay, so I have included a couple of exercises for you, but in reality our time is up and we are going to solve some of those in lesson 8. So at this point, I would like to thank you for your attention. Until next time.